Hello everyone, welcome to my presentation. I am Mohammad Zohurul Islam from School of Mechanical and Mechatronic Engineering. Today I am going to let you know the current updates of my research. The topic of my research is understanding the interaction of sterile drugs with model lung surfactant monolayers using molecular dynamics simulation under the direct supervision of Dr. Shubhar Shah and Dr. Evelyn Deplazes. Before going to talk about the current updates of my research, I would like to give a very brief overview about my original study plan. I started my research with focus on 4 particular studies. Study 1, the development of simplified lung surfactant monolayer models exposed to prednisolone for model and method validation. The second study was molecular dynamics study of different prednisolone drug concentrations to understand the effect of drug concentration on lung surfactant monolayer structure and drug diffusion. The third study was interaction of a ligand-coated gold nanoparticle on lung surfactant monolayer and its component at the air-water interface. And finally, the fourth study was on the effect of surfactant proteins and drug molecules in the interaction of ligand-coated gold nanoparticle with lung surfactant monolayer. In the next slide, I will discuss about why I have revised my original study plan and what modifications I have made. In this slide, I would like to talk about the reason why I revised my study 3 and study 4. In the middle of 2020, I found that one of the research group is working on nearly the same field, and they published a paper on ligand-coated gold nanoparticle, but they didn't consider the sterile drug in their study. In order to ensure the novelty of my research, I revised my study plan with particular focus on sterile drug interaction with lung surfactant monolayer instead of ligand-coated gold nanoparticles. And I also found that recent studies showed that steroid drugs, for example, prednisolone, dexamethasone, mometasone furoate, are currently used to treat COVID-19 disease, which also motivates us to work on this steroid drug interaction with lung surfactant. I am confident that the modified study plan will be novel, insightful, and innovative compared with previous study plan. Now I'd like to talk about my publication plan based on my study plan. I have made a plan to publish 3 Q1 journal papers from my PhD project. According to my publication plan, I submitted a paper in BBA biomembrane, which is currently under review based on my study plan 2. I have completed all the simulations and manuscript currently in preparation based on study 3. After that, I will start parameterization and simulation according to study 4. In the same time, I attended on national conference and on international conference. From them, a paper has been accepted for the publication in AIP conference proceeding. In this slide, I would like to talk about the overall progress of my study. In the first year, I completed 2 mandatory courses, Technology Research Preparation and Technology Research Method. In the same time, I completed the sufficient literature based on study 1 and submitted a conference paper which has been accepted for the publication on AIP conference proceeding. I have completed sufficient literature and model setup based on study 2. In the time of second year, I have submitted a journal paper and it is currently under review. The model setup and simulation have been completed for study 3. In the time of third year, I will submit a paper based on study 3 and I will finish all the procedures for study 4 and I will try to submit it in the journal. Here is the overall research plan. The colors refer to the progress that I have made so far and the proposed work that will be finished. I believe my progress is in right track and I am confident that I will be able to finish my project within this time frame. And finally, thank you all for listening to my presentation.